To another video for A-level biology for photosynthesis. Today we're going to be looking at the limiting factors of photosynthesis and what affects the rate of photosynthesis. Really important to understand this and be able to apply it to graphs and practical situations, which is where this is normally, where the questions, the higher level questions of photosynthesis will often apply. So where we're going to introduce this all came, started to be understood in 1905, where a scientist called Blackman introduced something called limiting factors. And basically as a result of the work, he formulated the idea of limiting factors. And the principle of limiting factors, or law of limiting factors, is that at any given moment, the rate of a physiological process is limited by the one factor in the shortest supply and by that factor alone. So what we're saying is if there's no carbon dioxide, it doesn't matter what the temperature is. If you've got a perfect temperature with no carbon dioxide, photosynthesis can't occur. So the limiting factor would be carbon dioxide. And you need to be referring to this as you explain your answers on limiting factors. So first of all, let's look at plants in the UK. So the optimum conditions in the UK, what would they be? Well, first of all, if we look at light, we want a high light intensity of certain wavelengths. So the more light, hopefully, just really basically, you should remember that light provides the photons of energy to cause photoionization and photolysis, and that means that you're giving those electrons the energy in photosystem 2, but also photosystem 1, and you're splitting the water molecule up as well. Now we can maximise those conditions in the UK by using glass houses in the day to get the sunlight in, especially in the summer, and then they can have artificial light on, UV lights on in the evening. Okay, and you can see here light intensity on this graph, and you can look at the fact, I'll look at graphs a bit closer with you later on, but as you increase the amount of light, the rate of photosynthesis increases to a certain level. We'll come back to the graphs later on. Temperature of about 25 degrees is the optimum for most plants. You can go up slightly higher, but it causes a strange side effect, which we'll talk about right at the end, which is an extension bit of information. But basically enzyme control reactions. So if the enzymes fall below 10 degrees, the reaction slows down, and if the enzymes go above 45 degrees, then the enzyme will denature. So it's not above 25 degrees the enzyme denature, it's 45 degrees. Okay, we'll talk about why 25 degrees is important right at the end. The stomata will also close to prevent water loss if you have too high a temperature, and that means that no carbon dioxide will get in. So I suppose that one links to 25 degrees as well. Plants are adapted in this country to be between 20 and 30 degrees. There's something called photorespiration, which is what I'll go into later on, over 25 degrees as well, which is a problem for a lot of plants. However, greenhouses or glass houses will trap heat, and that will maintain the temperatures, and you can also use heating and cooling system or air circulation if it's too hot. So then the other thing is obviously we need light intensity of certain wavelengths. So we said more light gives more wavelengths, so it gives more light energy, but the different types, the different pigments absorb different amounts of energy. So if you have a look at this graph, you can see that chlorophyll A absorbs most of its light at a lower wavelength into the violet light, and then again in the red light. Okay, chlorophyll B absorbs blue light best, and then it also absorbs quite a lot of red light, and carotene absorbs green light, okay, and none of them absorb yellow and orange that well. Okay, so that is your different absorptions. That's why in photosystems we have lots of different pigments which can absorb therefore as much of the light spectrum as possible. And there are other pigments, there are lots of accessory pigments, but we don't need to know them at all. So let's keep going then. So carbon dioxide, the concentration is 0.4% is optimum. Okay, in the air it's naturally 0.04%. So obviously if we increase that by a tenfold, it will become more particles to react to the ribulose biphosphate. And above 0.4, however, the stomata will also close, so it becomes a negative effect, which is interesting and shows you why carbon dioxide levels are vital. How can we increase the carbon dioxide levels? So we can burn small amounts of propane in a carbon dioxide generator, which is relatively easy to access and buy. Obviously, other conditions, we need water. Okay, too little, there is no photosynthesis. Okay, and too much, the waterlogged soil reduces mineral uptake. Now, water's a bit of a strange one because you never count it as a limiting factor in the exams, because technically, by the time it affects photosynthesis, the plant would already be dead if you didn't have enough water. So there are only really 3 limiting factors you're ever going to talk about: carbon dioxide concentration, temperature, and light intensity. So then let's have a look at this graph. Now this one's really important. So this shows you light intensity, and it's important you know how to read these graphs. So if you think about it logically, you could talk your way through this in an exam. So this graph is showing as light intensity increases, okay, until point X on the graph, as the light intensity increases, the rate increases. So what this is telling me is if I double my light intensity, I'm going to almost double my rate of photosynthesis, if not double it, okay? So at this point, until X, until it levels off over here, light intensity is the limiting factor. As I increase it, I increase the rate of photosynthesis. But then what happens after point X, I'm still increasing the light intensity because I'm still going this way along the line, but I'm not increasing my rate of photosynthesis. It's not going up anymore. So it doesn't matter if I keep increasing light, the rate of photosynthesis isn't increasing. And therefore, my rate of photosynthesis is no longer limited by light intensity. There must be something else. Now, I'll often present this as a graph like this one. Okay, so the second graph. And what we have here again is the same thing. So until about, let's say here-ish, okay, light intensity is the limiting factor. As I increase the light intensity, the rate of photosynthesis increases. But then it levels off, so in both graphs. So as I increase light intensity here, nothing happens. As I increase light intensity here, nothing else happens. So there must be something else limiting my rate of photosynthesis. Now you can look at this graph and you go, okay, well, let's have a look at this. Is it carbon dioxide and temperature? Well, the carbon dioxide is different between these two lines. So this would suggest that carbon dioxide could increase this graph, and it's the limiting factor on the red graph line, definitely. But if I increase the carbon dioxide to the maximum of 0.4% on this one, at this point, this graph is leveled off still, and I know if I increase carbon dioxide anymore from the previous slide, it won't go up. So at this point, it must be temperature that is limiting my rate of photosynthesis. Now, you'll often get asked what is the limiting factor. So if you got asked on this first graph, what is the limiting factor at X here, you would say either carbon dioxide concentration or temperature, because you don't know which one it is. And here we've got a graph, and again, as we look at this graph, as we increase the rate of concentrated carbon dioxide, the rate of photosynthesis increases until point X here, and at that point the graph levels off. So as I increase carbon dioxide levels still, there's no increase in the graph. So there must be another limiting factor, which would either be light or in temperature. And this graph again, you can see here at low light intensity, so at this point it's the light intensity is limiting this one, and this one is probably temperature up here because you've already got a high light intensity. So you might be asked to describe different answers linking to this. So you can see here, carbon dioxide added or not added affects the average plant height, and therefore carbon dioxide is a limiting factor. As you add carbon dioxide, the rate of growth increases. And here you've got a different version which just shows you the light intensity with different heaters and how that can affect it. So again, light intensity is your limiting factor on the electric heater graph until here, light intensity till here, and then it becomes something else. So then temperature. Now I mentioned this earlier on, temperature is obviously important. Temperature doesn't really affect the light dependent reaction because there are not many enzymes involved and the energy comes from light, okay? Temperature does affect the Kelvin cycle. Below 25 degrees C, as long as the temperature is increasing, it will raise the collision rate of enzymes and substrates to make more enzyme substrate complexes. So as you increase the temperature from say 10 to 20 degrees, it will double. Every 10 degrees, it doubles in rate of reaction. However, this is where we got to this earlier point. Above 25 degrees C, Rubisco actually changes how it works. It doesn't get denatured, but above 25 degrees C, its active site will start combining with ribulose biphosphate and oxygen rather than carbon dioxide, and that creates a process called photorespiration. Now, you're probably given information about this if you're asked about it in the exam. You don't need to know much about photorespiration, if anything, but basically this wastes the ruby P, it doesn't get remade, the carbon dioxide is actually emitted as a product, and I think some ATP as well, but we don't end up making any glucose and we end up wasting ruby P. And at higher temperatures, obviously more water starts being lost to the smart clothes as well. And obviously we then go further than 45 degrees, enzymes will continue to denature. But above 25 degrees, Rubisco actually starts accepting oxygen as a substrate rather than carbon dioxide, so it starts using up oxygen, not carbon dioxide, and therefore your products are going to change because you won't have enough carbons to complete the cycle, replenish the cycle, and make hexose sugars. So you might get a graph that looks like this. Now this is a higher level graph, but I think it's quite a nice question. So when you get a graph about RuBP, triose phosphate or glycerate-free phosphate, you've got to think about the Kelvin cycle. So if you look at this graph, it says what's the concentration of these, shows you the concentration of these in bright light and in dim light. So if you remember, the Kelvin cycle doesn't work for very long in dim light because the ATP supply will run out, and that's what this is sort of showing you. So let's just go and look at the Kelvin cycle for a second and just remind ourselves that in dark situations, when there's very little light, the ribulose biphosphate and the triose phosphate concentrations are going to drop, okay, and the glycerate-free phosphate is going to increase when the light disappears. Why is that the case? Well, let's have a look at this and it will explain it to us, okay? So if you look at it, ribulose biphosphate and triose phosphate. So if we start with GP up in the right-hand side, to get to triose phosphate, you need ATP, which can be used up, and you need NADP. The supply of those is coming from the light dependent stage. So if you've got no light, you run out of NADP, reduced NADP, and you run out of ATP. So if there's no light, very quickly that will run out. And that makes sense then that our amount of triose phosphate will drop when it is no light, and when there's lots of light it makes it regularly. And RuBP also drop because again you're losing your supply of ATP because there's no light from the fiber membranes, so therefore your RuBP will drop. But what will happen is any RuBP that's present in the stroma will still be able to be converted to triose phosphate, because that glycerate phosphate, because that doesn't require any energy to do so. So that means your levels of that will increase and then it will stall. It can't be converted into triose phosphate, so that will increase. So look at that written down. So in bright light, you have increased levels of independent reaction because there's more ATP and there's more energy provided from the light dependent stage, because more electrons are getting excited and therefore you're getting the energy. There's more NADP and ATP made as a result, or more reduced NADP, and that means your glycerate-free phosphate is converted to triose phosphate. In the dim light, you've got decreased levels of the reaction because less ATP is generated in the light dependent stage, and GP cannot be converted into TP or ribulose biphosphate, and therefore your GP levels build up as a result because they're being stored and they can't move any further into the other ones. So then you might have a question like this: well, what if you've got the same light level but you've got different carbon dioxide levels? So be careful with this one, because you look at this one quickly and get your carbon dioxide levels wrong. 0.04 is a high concentration of carbon dioxide. 0.008 is a lower concentration. So if you've got a higher concentration of carbon dioxide, you will have more GP and TP, and you will have less ribulose biphosphate. Whereas if the carbon dioxide levels drop, you get more ribulose biphosphate and you get less GP and TP. So let's have a look at why. So if you think about this, if you've got high levels of carbon dioxide, as soon as you get RuBP, it will combine with the carbon dioxide, and that will mean you get more GP and more TP. Okay, but if your carbon dioxide levels are low, and that means that if they're, if they're below a certain point, the ribulose biphosphate won't have carbon dioxide to combine with, and therefore it'll start building up around here. And that means that your GP and TP, because remember you've got lots of ATP and NADP, is not worrying, not a problem, that will continue to push any of them back there. So you'll end up slowing down like a traffic jam. It won't, because less carbon dioxide, you won't get this bit happening to replenish the RuBP, sorry, to replenish the GP and TP. So how do we answer this in a graph? Something like this. High carbon dioxide levels means more carbon is fixed into the carbon cycle. This means more GP is made, and that's converted to TP. The RuBP is generated, but it is used almost instantly into triose phosphate or into glycerate-free phosphate and then triose phosphate. At low carbon dioxide levels, less carbon enters the carbon cycle, and that reduces the rate of photosynthesis. And that means that you end up with RuBP is less likely to be converted into GP. So the RuBP will start building up, and the GP and the TP will be made less frequently, less rate. Temperature, as we said, temperature has little effect on the light dependent reaction, okay, but the light independent reaction, we just said about this idea of 25 degrees or above, it becomes what we call an oxygenase enzyme. Again, you don't really need to know this, but it could be asked in an exam and you might be able to analyse it. Photorespiration rates, when they exceed photosynthesis overall, you end up wasting ATP and NADP, so it's no good for anything really. Okay, the enzymes also can get damaged if you go above 45 degrees. So I hope that's been useful for you guys. I'll see you again soon.